The screen will not be visible. Please can be shared. As long as I can come in here somehow. Hello, everyone. Welcome everyone who's here. Welcome online audience. I hope you can hear us. If you cannot hear us, see us, please put it on the chat. That's the only way we can communicate with you. If you're watching on Zoom, if you're watching on YouTube, please put your questions and comments in the chat. We have people looking at the chat. We will try and take your questions either during the talk or at the end of the presentation. Today is a very special fifth Sunday Chai and Y. Our fifth Sunday's Chai and Y is usually online, but today, 30th October, also happens to be TIFR's Founders Day. So Shravani, you're doing a great special Chai and Y on Founders Day, and we actually are doing it live from a room in TIFR here. So before we get into the talk, Shravani, we must. Do you want to try and use the Bluetooth mic, or are you going to be loud enough so that people can hear you?
I think I'll be loud enough.
Okay, so online audience, could you hear Shravani? Can you hear Shravani in the back? Yes, you can. Okay, just shout.
Yeah.
Okay, good. So we won't worry about changing mics. We were doing everything last minute. Is low? It's low. Calvin says it's low. Okay. Put that. Turn it on. Turn it on. Turn it on. Just give us a while. We will change to the. Okay, just give it on. Meanwhile, let me introduce our speaker for today. Shravani, come to the field of view, please. Yes. Shravani is a research scholar in the Department of Biological Sciences at Bluetooth on. Not my laptop. Okay, our Bluetooth is not on, so we have to get Bluetooth on somehow. Where do you get Bluetooth on? Can you just turn the Bluetooth on? Meanwhile, Shravani, why don't we introduce yourself? Yeah, so she did a master's in bioinformatics from in Bangalore, and then she came here to do a PhD. She works in the lab of developmental biology and works on and cell biology. Okay, now we are add a device, tap to set up your P2 BT. It's connected. Are you sure? P2 BT would like to pair. Now? Yeah. Now, connection successful. Okay, let's go back to Zoom. Let's choose. It doesn't show up here. No. It's connected, but it's not showing up in Zoom. Connected. Headset. Okay. Now, Shravani, will you speak and see if this is better?
Hello, everyone. Can you hear me?
Audience, can you hear her? Online audience, can you say something if this is better? If you can't hear me, it's okay. I'm going to be going out in a bit, but most speak.
Yeah. Hi, everyone. Can anybody hear me? Yeah.
Much better. Somebody says much better. Thank you so much.
Thank you.
Okay. With that, you're going to tell us something about DNA, reading the code, sequencing, breakthrough prize, all yours. We're going to sit and listen, have fun.
Yeah.
Okay,
sure.
Online audience, remember to ask your questions.
So first of all, I would like to thank Arnab for the good introduction. And now I will start my talk. So basically my talk deals with
Yeah, height floating. Yeah.
Okay. So let me start again. Hello, everyone. So basically today I'll be talking about DNA code. What exactly do we mean by DNA code? How do we actually read this DNA code and understand it? And what kind of applications can we get based on this DNA code? Why is it important to study this DNA code, and how does it help us humans to actually make our lives better? So to start off with, I think, okay, to start with, everyone knows that the basic unit of life is cell, right? I mean, that's something that we have been reading and hearing about a lot. So even though we say that the basic unit of life is cell, how does a cell lead to a formation of these organisms? And that's where hierarchy of life comes, where different cells combine together and function to form a tissue. Tissues combine together to form an organ system. Now the different organ systems actually function together and work in coordination with each other to form a fully functional organism like ourselves. So even though we say cell is a basic unit and it's the unit that actually leads to a functional organism, what in cell actually leads to these kind of specific functions? Each cell functions very differently, and all these cells need to coordinate among themselves to form a fully functional organism. So what in cell actually leads to? So that is nothing but proteins. So there are different kinds of proteins present inside the cell. They are of different shapes, sizes, and the amount of protein itself is different in different time points as well as in different cells. It is said that if you actually generate the type of proteins that are required at the right time, in the right place, in the right amount, you can actually generate a functional organism. So that's how proteins act as a functional units of the cell. But proteins should also be made of something, right? They are not present as molecules, a bigger molecule, but something else makes these molecules. So that is nothing but amino acids. So amino acids are these small molecules which are around 20 in number inside our humans, and these are of different shapes. So you can imagine something like Lego. So for example, if you take any kind of, I mean, we all play with Legos when we are kids. So these are of different shapes and sizes. And when you combine one Lego over the other, you can create these lots of amounts of different structures that are there. So in the same way, these 20 amino acids, which are of different shapes and sizes, can actually combine together and form this chain of polypeptides, which can then be folded in different ways in order to produce these proteins. And depending on the amino acids number, the type of amino acids and the sequence of amino acids together can give us these different kinds of proteins that we see inside the cell. So how do we know that what kind of amino acids needs to be placed, where they need to be placed, and how do we fold them in order to make these proteins? Where is this information coded? And how does cell know that, okay, this is the time I need to produce this protein, and this protein requires this sequence of amino acids? That information is encoded in DNA. So DNA is basically deoxyribonucleic acid. It is small atoms that are combined together to form this double helical structure, which is the DNA. And just like to create any kind of structure, such as buildings or cars or any kind of structures per se, we need particular blueprint, like which tells us the information as to how or what kind of material is necessary, how much is the dimensions of it, and what kind of information is required before actually starting to build the structure. So in the same way, DNA stores this information and hence is called as the blueprint of life. So basically, it stores all the information that is necessary in order to produce the organism. And these organisms can range from the smallest of bacteria to the largest of us, that is more complicated humans. So like this, DNA acts as a blueprint of life. So we do have DNA, but what exactly is this DNA? So this is more of a simplified version of DNA, where there are two strands which are placed together in a double helix fashion to form this DNA. So if we take this DNA and look at it, it's like a step ladder where each step is comprised of a molecule called as nucleotide. Now in this nucleotide, if we see, there are three major parts. There is a phosphate group, there is a sugar group, and the base, as you can see over here. So now this sugar and phosphate together combine, and when one nucleotide and other nucleotide interact with each other and form this step ladder type formation, these sugar and phosphate together form the backbone of the DNA. And the base, there are basically four different bases, that is adenine, thymine, guanine, and cytosine. And all these four bases here can be represented as different colors and the letters, as you can see, A, G, T, C. And these form the basis of giving the information that is there. So when you take a single strand of DNA, the sequence that you read out, which is A, G, T, C, A, T, G, A, like as you can see over here, is basically the DNA sequence, which encodes the information that is required to form the proteins and thereby the functional organism. It's also important to know that when one strand of DNA has a particular base, there is another opposite base that is present in the other strand, which is called the complementarity principle. So whenever there's A on one strand, the thymidine or T acts on the other side. And the same way, if there is a guanine, there is a cytosine on the other side. And this acts vice versa as well. So these bonds are necessary to create this double-stranded model, and this makes up the DNA, which helps in the storage of information. Now, if we consider the information, the amount of proteins or the information that is coded, it's millions and billions of base pairs long. It is so long that it is said that when you take out DNA from all the cells inside the human body and lay them side by side and extend them, it occupies a whopping distance of 744 million miles. It is such long distance that if you consider traveling from Earth to Moon, it covers the distance around 1500 times front and back. So it's that long. Now, but when we consider cell, it's very small, like it's very small basic unit. So how is this DNA present? So this DNA is actually packaged into smaller and compact structure called as chromosome, which is present inside the nucleus. But DNA is present inside the nucleus, yes. Proteins are synthesized in cytoplasm. Now, how is this information translated from DNA to protein? That is a bigger question. And that's where the process of central dogma comes into play. Here, what does DNA do is that the cell cleverly actually generates a small copy of DNA, which is called the RNA or the messenger RNA. And this acts as a messenger which carries the information, whatever is present on the DNA, as a smaller fragment into the cytoplasm, where other proteins can actually recognize this sequence and can attach the amino acid that is necessary to form the protein. Now, this process of translating information from DNA to RNA to proteins is called as the central dogma of life. Now, how is this information actually translated? Now, when I said the code, DNA code is just ATG, which is just 4 letters which are arranged in different fashion. Now, how is this read to make an amino acid? So the protein that is present in the cytoplasm reads the code in 3 at a time. So for example, as you can see here, ATG, which is generally translated as AUG in the messenger RNA, codes for one particular amino acid. Like that, it keeps on reading 3 bases at one at a time, which code for specific amino acids. And thereby it knows what amino acid to add one after the other in order to make a functional protein. This is just a brief to show you how it looks, that how amino acids are coded by different DNA bases. So each amino acid, since there are only 20 amino acids, each amino acid can be coded for more than one codon or the 3 base letter that we see, 3 letter code that we see. So, okay, now we know why it's important to study.
I'm sorry, I didn't understand the last figure.
Yes.
Can you repeat the question and explain what this figure is?
Yes. So as I said, the 3-letter code gives an amino acid, right? So now exactly, now we know that there are 20 amino acids and there are 4 letters that are present. Now if we take into combinations of the 3 letters, there are many possibilities that come into play. Now there are only 20 amino acids. So each amino acid actually codes for more than one codon. So for example, if we take anything such as valine, it can have U compulsion, but can have other codons present. For example, there are many stop codons. The start codon is AUG, whereas the stop codons are UGA, UCA, and UAA, which all of them act as a stop codon in order to stop the formation of the protein itself. Like that, each amino acid can have more than one 3-letter code, which can code for itself. Yeah. Oh, sorry. Yes. So, okay. So the question is basically that I've been speaking about T till now, ATGC as a code, but I've suddenly mentioned U. So what exactly is U? So whenever we translate information from DNA to RNA, the T or the thymidine gets converted into uracil. And this is the U that I've been speaking about. And this uracil, together with other bases, can be recognized by the proteins to form the amino acids. Okay. So yeah, we have spoken about the DNA, the importance of DNA, and understanding the DNA code. Now, DNA codes for these proteins, and hence it's important to understand what is the sequence of codes or the base DNA code itself to understand how the proteins are generated and how the cell functions itself. Now, how can we see the DNA? Like what is the methods that can be used to study this code of DNA, and how do we see these? So when I say DNA code, though I keep saying that it's ATGC, which is just 4 letters for us, but it's actually chemicals that are there. So using this property of chemicals, that is, since they are chemicals, they can be reacting with other molecules to produce different things. So using this idea, one of the scientists called Frederick Sanger has discovered a method to identify how the bases are there or arranged one after the other, called the DNA sequence. And this method is called as DNA sequencing. So he has won a Nobel Prize for identifying this method of DNA sequencing. And this method was developed basically in 1977. And this method has been used for around 40 years, where he used a modified type of nucleotide, not the normal nucleotide that we have. So I'll be explaining what exactly he has done. So when we consider a normal nucleotide, so imagine, as I said, it's a step ladder type structure where one nucleotide adds on to another nucleotide to form the strand of DNA. Now, when we consider a normal nucleotide, it has molecules which, when interacted with other nucleotides, can extend itself to form the single strand. But if we modify the molecule or the atom which is required for the interaction, we actually terminate the sequence. So for example, in this DNTP, this OH group is actually necessary to add on the other nucleotide onto itself to extend the chain. But when we replace this OH with a hydrogen atom, we see that the extension cannot happen, and thereby we lead to termination of it, or basically stop how the DNA is extended. Using this interesting method, Sanger developed a method of sequencing it. So he used a mixture of these normal nucleotides plus the nucleotides which are modified, and each type of nucleotide, that is either A, T, G, C, each is bound with a particular fluorescent molecule. Now, when this mixture is added to a single strand of DNA and they introduce a small primer, basically a short strand of DNA, which can actually bind to the DNA strand and can help in extension of one nucleotide at a time. So when we introduce this and introduce all the modified as well as normal nucleotides, we see that they can attach to each other randomly, like based on the sequence that is present on the DNA strand that is there. They can add either a normal nucleotide or add a modified nucleotide. Yes. Yes. So yes. So the question is that whether this addition of nucleotides occurs normally inside the cell. So yes, the enzymes are present inside the cell, which actually help in increase or adding more nucleotides to the DNA. Technically, only during replication it adds to the DNA, but in normal conditions it won't. Whereas again, in RNA or anything, then again these addition of nucleotides adds upon each other in order to produce this messenger RNA. Okay, so as I was explaining, now we have DNA strand, a single strand of DNA. So since it's double stranded, we remove one. Since it's double stranded, we remove one of the strand of DNA and we try to sequence only one strand, because the complementarity will help us understand what base is present on the original strand. So now the single strand of DNA, along with the mixture of modified versus unmodified nucleotides, are added together. And now upon random inclusion, whether based on the complementarity, it can either include a normal nucleotide or a modified nucleotide. If a normal nucleotide is added, then it will continue the elongation. But if a non-modified nucleotide is added, it will stop the reaction there itself, forming a smaller fragment of DNA. Now, once this reaction continues for some time, we generate like multiple different fragments of DNA, which are of different sizes. So these sizes can be dissolved under a gel, where based on the size of the fragment, you can identify which position is it at. For example, as you can see over here, so these are the different sizes of fragment. And as we know, like if the fragment size is smaller, it will run faster on the gel. So it's more on the downside of the gel. Whereas if it's larger or bigger, it takes more time to go through the gel itself. Yes. So this is basically, I'm sorry. Yes. So this is a polyacrylamide gel. Not going into complete details of it, it has molecules which are like mesh-like patterns. So now this mesh, when forms, it has holes in it which can help pass these small molecules. And so these have holes which are there, and these whenever you imagine a small thing with a lot of barriers around it. Now, whenever it's very small or thin, it can actually go through all the gaps and reach faster. Whereas someone who is bigger and larger might take slightly more time to go through the holes to reach over to the ending point. So based on the size, there is a resolution or change in the amount of distance that a particular strand can travel. So as the smaller fragment can be at the last of the gel, and based on the fluorescence that is present on the nucleotide, you can identify what is the sequence of DNA. So here the T, which is the smallest, is present over at the end of the gel. And just by seeing the color and the position inside the gel, you can actually read out the sequence, like T, C, G, A, G, T, C, A, G. So basically, this is the DNA sequence that we talk about. And this is a method that has been introduced by Sanger. The development has occurred, of course, and now people have started using, instead of gels, machines, computer machines, which can actually detect the fluorescent signal, where when passed through it, it can resolve based on the size, the fragment, and can detect the signal directly by using computers. So this method of Sanger has been very crucial for us to actually identify the human genome. One of the biggest breakthrough in biology and a big project that has been taken up by US Department of Energy and National Institute of Health, they came up with this idea that using this method, can we actually sequence the whole genome of our humans? Now, so this led to the introduction of the project, Human Genome Project. So it started in 1990, and it took almost 13 years to actually sequence the whole human genome. So the basic goal of this project was to sequence the whole genome, to understand and get a physical map, like to know where exactly what DNA sequence is present and what kind of genes would it encode, to actually understand the functionality of the different genes, and to use this information in a case when there is some difference in the code. So we know that whenever there is a difference in the code or whenever there's a change in the code, it can lead to drastic results with respect to proteins and can lead to many diseases. So having this kind of a reference where we know what exactly is the sequence and then comparing it with other things will help us understand what are the changes that have occurred throughout the evolution. So this Human Genome Project was one of the biggest feat of biology, which helped in understanding this human genome, and thereby had made research more easier and more understanding, and made us ask more questions. So how did they do it? They extracted the whole DNA. They have taken around 20 volunteers, and they have taken the whole DNA of those from the cells. They have fragmented into slightly smaller pieces. And now to actually, as I said, the DNA is stored in the form of chromosomes, and there are many chromosomes. So to make a physical map of it, to understand what part came from which part of the chromosome, they are first made into longer fragments, which are cloned into the bacteria and stored aside. It's basically a record keeping type. Like to actually know which comes from where, we have taken it from there and put a record that, okay, this fragment has come from here. Now, whenever I require to sequence, I can take out this fragment, again fragment it and sequence it. And now I know that, okay, this is basically coming from this region of the gene. And now if I want to combine it with another side-by-side gene or the sequence, I can easily know because I already have a record of what is going on. So like this, yes. Yes. okay, the question is basically when we are cloning into the bacteria, do we know which region comes from which part of the chromosome? So we exactly don't know whether, as in side by side, we don't know, but as a whole, from which chromosome is it coming, that information is stored. So this helps us make our life a bit easier. Yes. Yeah, I mean, we do know from which chromosome or which part of the chromosome did we isolate the DNA. So we do have a basic idea as to which part has it come from. Since we do not have a reference right now, this is basically the reference generation. Yes. And to generate a reference where everything is directly broken down and then put together becomes difficult, because as I said, DNA is very, very long. And so many fragments assembling together, and it's only 4-letter codes. There can be repeats and stuff, which makes it much more difficult to put them together directly. So rather than that, parts of it can be taken out, and which can keep as a record that this is coming after this. So have a basic idea, though it might not be as continuous as I said, but it can have some gaps which can be further filled out by other methods of molecular technique. So that's how they generally has done. Okay. So now that we have seen that it has a bigger impact, how exactly is Sanger sequencing helping? Whether as any technique in the world, it has its own advantages as well as disadvantages. Now, one of the major advantages of Sanger sequencing is that since it tries to code one base at a time, it is more accurate. That is, the errors in coding is very less. And this helps in getting the DNA sequence very precisely and efficiently. And it generates a reads which are longer. So when you try to sequence this big of a genome, you want something which is more larger fragments rather than smaller fragments that are there. So having a slightly larger fragment will help us to combine those fragments more easily to form the bigger fragment. So one of the advantages is generating bigger fragments. But there are also a lot of disadvantages associated with this method. Some of the major disadvantages is that the cost. So since it requires a lot of process of cloning and then recognizing the sequence of the DNA, the cost that is required for this is very, very high. And the time that is required to generate this whole sequence is very long. Since again, there are multiple steps involved, so you take a lot more time to get the DNA sequence. So hence, this actually prompted us to bring out more sequencing techniques or different other sequencing techniques, which can improve upon these factors to make our life easier and to sequence genomes much more faster and with less errors and more easily with less cost. So now this started the era of next generation sequencing. So before understanding how this actually revolutionized in such a way that now we can sequence any human very fastly, we need to first understand what is the history of genomic sequencing. So as I said earlier, the Sanger method of sequencing has been developed in 1977, followed by which this method has been used to start the project of human genome in 1999, 1990, and which extended up to 2003. Between these times, they have also generated the genome sequences of different model organisms. So just now, as a lot of people here in TFR might have looked into different organisms such as C. elegans, flies, and zebrafish, which are basically small organisms that can help us study the human development or any kind of processes in humans much better. Since we can't do things or manipulate things directly on humans, we use these model organisms as a substitute and understand the things that are happening inside the cell better. So the genome sequences of these different model organisms has also been generated in this mid-time using Sanger sequencing. But as I said, since it requires a lot of time and money and the techniques keep on developing, so this led to the era of next generation sequencing, where different type of sequencing technologies have been developed. And we are at a stage where now we can sequence using a sequencer which just fits in the palm of our hand. We are at that stage, which I'll be speaking slightly later. But to first go with what kind of techniques have been developed and what kind of modifications have been done to make it easier and better to sequence the genome. So the basic methodology is slightly similar to what we have seen earlier, but there are slight tweaks that have been done which makes it more efficient. So as any other technique, you isolate the DNA and you fragment it into smaller pieces so that the reading becomes more easier. Now you attach small known sequences to it, which help in amplifying. So I'll come to the amplification step. So basically, whenever you have a single copy of any code or anything, go into it if there are multiple people who wants to solve the code. If we have only one copy of it, it becomes difficult because everyone needs to wait for their own turn to get the code and then read the code and analyze the code. But if we can make actually a copy of that code and give the same code to everyone, then they can sequence or they can understand the code each individually and can generate it faster and can solve it faster. So in the same way, we use DNA fragments and we amplify the fragments. That is basically make multiple copies of it using either a small flow cell or basically any straight platform, which is micro platform, and we generate multiple copies of it of the same sequence of DNA to form a cluster. Now, so each cluster can give out a single signal. So if it's only one fragment, the signal that we get is very low. But when we have multiple fragments that are present, the signal can be amplified and can be increased to multifold. So thereby can help in detection of the signal very easily. So this is called as a cluster formation. And once we get the cluster formation, we can actually sequence them. I will be explaining the different methods in which we sequence it and finally assemble it to get the actual sequence of the DNA. Now, one of the most common method that is used for sequencing is Illumina sequencing or sequencing by synthesis. So this kind of uses the knowledge that we got from Sanger, which is basically using fluorescent nucleotides, but they do not use a termination-based method. Here, what they do is they attach again a single fluorescent molecule to the nucleotide, and each nucleotide is with a different fluorescent molecule. Now, whenever it goes and attaches to the DNA or the complementary base goes and attaches to the strand, growing strand, we see that as soon as it attaches, the fluorescent molecule is cleaved and released. Now, whenever this attachment occurs and the release of the fluorescent molecule is there, this can be visualized by us or basically the machines, and they can detect what color of fluorescence has been released. And by using the color that is produced, you know what kind of base has gone and joined to it to make the DNA. Like that, you read from each cluster what are the colors that are attaching or that are being released at each step. So it's the images for the fluorescence are taken every second, and now you just see the change in the color and you detect what color it is and make the sequence by yourself. So yes, yeah. So for example, in this image, as you can see, these are the shots of basically you take multiple shots of it one by one and see what colors are formed. So as you can see here, like at the same position of the cluster, you have different colors forming, and each color represents a particular. So just by reading it like this, like green represents C, blue represents G, C, T, A, C. So like by just reading frame by frame and seeing the color that is present, you actually know the sequence of the DNA, and thereby makes it easier to understand the DNA sequence. Yes. It's just multiple, yeah. So the question is, what is happening? What is 1, 2, 3, 4, 5, 6? So as I said, one base addition produces one fluorescent molecule. Now, as soon as it is gone, the second nucleotide is coming and another fluorescent molecule is produced. Now you need images such that it catches the difference between first fluorescence emission versus the second fluorescence emission. So you continuously keep on snapping images and see what exactly the fluorescence changes are occurring. Now, by taking this continuous snapping, you know the changes that are happening in real time, and thereby you know exactly what base is added one after the other. Yes. So there are methods in which initial methods used a single base as well, but in the later stages, they have efficient tools which can detect the fluorescence very quickly. So they use all the nucleotides together and then snap the image. Yeah. Sorry? Yes. No. So see, as you see in this flow set, okay. Yeah, I'm sorry. So the question was that if I'm adding only one base at a time, or I'm adding all the bases at a single time. So if you see, this whole square is basically the region where different clusters are formed. Basically, different DNA sequence, shorter fragments are attached to this region, and each DNA fragment forms a single cluster. Now here, each dot represents a different DNA sequence. So now when you flood this area with all the fluorescent molecules of different nucleotides, so depending on the sequence, the nucleotide can go and bind to one cluster, and a different nucleotide can bind to different clusters. So for example, in this cluster, there is T, whereas in this cluster, there is A, as you can see over here. So since it's flooded, depending on the complementarity, the different base can go and add itself to the cluster by giving so this is what makes this technique more efficient. That is, you can actually sequence a lot of DNA sequences at the same time. Hence, it's called as massively parallel sequence. That is at the same time. Yeah. Yes. No. Yes. Yeah. Sorry, please continue. Yeah. Yes. So if you add all four, the question is, it will be taking on continuously, and hence you might see more than one fluorescence. But that's the efficiency that we see about. That is, we are taking snapshots at each second. I mean, how the process is occurring, and just the release is what we are seeing. Yes. Yes. Yeah. So basically, okay, the question is, no, not necessary. So that's what. It's just, yeah, I can discuss personally with you and let you know what. Yes. Okay. So one other method that people have developed is pyrosequencing, which uses the light as a model to detect the signal. So here what they use is basically, since I said it's a chemical reaction, when a nucleotide is added, it releases a lot of byproducts. Now, one of these byproducts can actually be interacting with other enzymes to produce light. And based on the signal of the light, you know what base is added. So in this technique, you add each base separately and not together. So whenever if the correct base is added, the light is produced. And whenever the wrong base is added, the light is not produced. As you can see in this example, for example, in this DNA sequence, the base over here is cytosine. And as we know, the complementarity is G or guanine. So now whenever other letters are added, for example, A or T, the light doesn't get produced. But whenever we add G, there is a chemiluminescence or light that is produced. And now we know that, okay, this position is occupied by G, which is complementarity to C. Hence, the DNA sequence has a C in that position. So like that, pyrosequencing can be used to produce the DNA sequence. Another technique which uses slightly similar kind of a mechanism, that is, it uses the byproducts itself. But in this, instead of using light as a mechanism to detect, they use voltage changes to detect. So the byproduct is basically hydrogen, and whenever the hydrogen interacts with the solution that is present, it can create voltage fluctuation. And here also you add one base at a time, and if hydrogen is released or is there a voltage fluctuation at that time, we see that the voltage is depending on the voltage difference. We can say that, okay, this is the nucleotide that's added. And based on the amount of voltage difference, we can say whether it's two bases that are added which are of same type at the same time. So that's how you can detect the sequence of the DNA using this method of ion semiconductor sequence. The last method that I'll be discussing about is sequence by ligation. This is slightly different and a bit more complicated or different than the methods that we have till now seen. So here the mechanism that is used is basically ligase, which is a molecular glue. So ligase is it's like a pasting molecule that is present inside the cell. So whenever there is a break in the DNA strand or whenever there is some kind of basically any break in the DNA strand, this molecule comes, it sits, and it joins these two strands together to form a single strand. So here they use a slightly different method where, yes, the question is basically, can we call the ligase a glue? So as I said earlier also, ligase is a molecular glue. So it's not typically like what we say, but since it attaches DNA strands together, it is called as a molecular glue or as referenced as molecular glue. Okay, so in this method, they use a short strand of DNA itself again, and instead of detecting single base at a time, they detect two bases at once. So they generate this small strand of DNA where the first two bases are the bases which we know, or what is exactly the base. And this sequence is attached to a particular fluorescent molecule which fluoresces when cleaved. So whenever it attaches and there is some kind of cleavage from the sequence, then it fluoresces. So what they do is they attach the sequence of it to the solution containing the DNA strand and the primer, and they add this ligase or the molecular glue. Now, if there is complementarity between the DNA sequence and the actual DNA strand, this short strand of DNA can actually ligate itself or attach itself to the primer. So whenever this attachment occurs, the fluorescent molecule can be released and can be detected by us, which again tells us what are the two bases that are actually complementary to the DNA sequence. Once it's run for a certain amount of time, the primer that is used as an initial sequence as or a starting point is shifted by one or two bases so that it can be read all over again. So like this, every base is read twice and we can understand what exactly is the sequence of DNA. Okay, so now we have discussed so many different methods of DNA sequencing. I mean, there are quite a few techniques, but why do we need so many techniques? And is there any advantage to have so many techniques? And how do we differentiate which to use when? And why to use a particular sequencing method over the other sequencing method? So it depends on the kind of question that you are asking. For example, if you are trying to generate or sequence a very long fragment of DNA, you want something which generates reads which are slightly longer. That is basically the fragments which are longer, so that it becomes easier to attach these bigger fragments to form a single fragment. So to generate these kind of bigger reads without any errors, it's better to use light-based sequencing over other kind of sequencing methods. Now, in another case, you might want to get the results fast. Rather than looking for the size of it, you want to sequence a small part of the DNA, especially in some kind of a disease condition or something. You want a quick result to know what kind of treatment to give or how do we deal with the particular disease. And in these conditions, you want the quicker result. So you would go for a technique which is more efficient with respect to time, that is how fast it can sequence rather than going with how long the reads are there. So like this, different sequencing methods can be used in different conditions to give the results that we want. So now that we have generated so much data, as I said, when we consider whole DNA, it has high millions of billions of reads that are generated. And one of the major problems that we are facing right now is this high amount of data that is present, and we do not have the capacity to actually process and analyze this data more efficiently. But there are still some methods that have been generated which can be used to assemble this gene. Now, since these reads are there, these are like puzzle pieces. Now, these puzzle pieces needs to be put together to make the proper puzzle which actually makes sense. So now, just like it can be done in two ways, basically resequencing and de novo. So imagine you have a puzzle pieces, but you already know how the puzzle looks like. So now it's easier to attach the pieces since you already have a reference. You know which place goes, which piece of the puzzle goes where, and you can just compare to it and attach the pieces to form the whole puzzle. That is basically resequencing, where we already have a reference genome and we take these reads and see where they actually overlap with each other and then combine them to form a single strand of DNA. Whereas de novo assembly is like the puzzle pieces are there, but we have no idea what exactly is the final picture. So what we do is we try to look at the shape of the piece, we try to look at the color, the design that is present on the puzzle piece, and we combine all these pieces to create a meaningful puzzle. So in the same way, in de novo assembly, we rather look for the overlapping regions. So when we have two different reads and there is small overlapping region, we can actually combine these reads and bring up a whole sequence. Now, each has its own advantage, as I said, and is used in different places. Now, if you all just want to detect the changes in the sequence, like for example, though we are all humans, we look very different, and there are certain differences that are encoded in the DNA sequence itself, and you just want to identify these sequences. So what you do is you just compare one sequence with the other sequence or the reference genome. And the whole genome project has already given us a reference genome, which can be used as a reference and then can see what kind of changes are occurring in our code and what kind of changes are there in the DNA. In that case, you use resequencing method. But when you want to sequence a new organism, like we have identified a new organism, we know nothing about the sequence of the DNA, and you want to sequence the whole DNA to actually see the functionality and understand the DNA sequence and thereby the proteins and the codes and all this stuff, then you use de novo assembly, where with just by read assembly, you create the whole DNA sequence. So as we have progressed, the cost for sequencing has reduced a lot, and by developing these new techniques and reducing increase in the technology advances, the cost per base sequence has reduced a lot, and hence makes utilization of these sequencing methods very high. We can use it for so many different purposes and to answer so many questions. For example, it can be used to understand in any disease conditions what exactly is the change in the code that is happening that is leading to the disease. Or in a normal cell development, one cell is functioning differently versus another cell is functioning differently. It can be a change in the DNA code, or it can be a change in the messenger RNA itself that is actually transferring this information. In both these conditions also, you can use the sequencing methods to actually determine the sequence and thereby understand the difference. Apart from that, if a particular population is coming from a particular area, based on the environment, there are certain changes in the DNA that occur. And these changes can also be detected by the DNA sequencing, and therefore you can understand the ancestry that is present in yourself or where you originate from. So like that also it helps. And finally, NGS sequencing is not only used for DNA as I've been speaking, but can also be used to code the RNA itself. So by understanding the messenger that is present and the number of messengers, the type of messengers that are present in the each cell, we can answer many questions as to the functionality and what are the changes that are actually leading that is making the cell behave differently from the normal cell or in its original state. So this actually has led us to something called as precision medicine. So as I said, humans behave differently for different things. For example, a person who's living in a colder conditions will behave normally or can adjust properly to the colder conditions very easily. But a person who has been living in a hotter conditions might take more time or more difficulty in adjusting this. In the same way, the environment can actually impact DNA sequence, as I've said earlier. Also, when we consider any kind of diseases, now the disease might be a single disease, but the root cause of the disease can be very different. It can be due to the change in DNA code, which can be of different types, or it can be change in the messengers that are actually transferring the information, or it can be the change in the protein itself that is encoded. Now, though we say the disease is the same, the treatment that we are giving is also same till now. But when we see that the root cause is different, isn't it better to actually treat the disease based on the root cause and give a more personalized medicine so that we can improve the life status a lot more? So that's where NGS comes into play. NGS can actually help in sequencing each individual and thereby can give more targeted medicine and can make efficient living. So this is just an example of a study where people have sequenced the individuals which are dealing with these particular diseases and have identified different root causes or the change in the nucleotide code that is actually leading to this. Especially in rare diseases cases where we actually don't know the root cause, the NGS sequencing helps very highly where you can actually detect the change in the genetic code itself. So thereby helping in giving treatments and increasing the efficiency of the living organ. So finally, I'll end my talk with a small advancement in the sequencing technology itself. This is something that I've mentioned earlier, that now the sequencing has improved so much that now you can sequence using a sequencing machine on the palm of your hand. So this is Million, which is a sequencing machine that is so small that you can carry it anywhere. And it just requires a small amount of genomic DNA. So you can actually sequence anywhere. Like you can go to a rainforest, take up a particular sample and even sequence. Or if you are sitting inside the lab, you want a quick result, you can do it. So this makes sequencing much efficient and more easy to do. So this work would not be possible with the greatest minds of science who have actually developed these techniques and have helped us to get to know this DNA sequences. These are the three scientists, Shankar Balasubramaniam, David Kleinerman, and Pascal Mayer, who have made contributions in generating these techniques and thereby have received the Breakthrough Life Science Prize this year for their greatest contribution. Yeah, thank you, and I'm happy to take some questions.
Thanks. We will talk to everyone here, but let me just go here so I can pick up your mic.
Yes.
There are a few questions in the chat. So Urmi is asking, is there any method or software which can store the whole human genome sequence?
So they are bigger computers and memory storages that have been developed through the course of years. But this is something that is still an issue, that when you actually take up whole genomes and of different organisms, storing this kind of big data is actually very difficult. And especially processing it to make it assemble itself and to run this amount of data becomes very difficult. But there are machines which store these data. They are gigabytes of information which can be still stored. Yes.
Okay, and there's another question which asks, using all these methods, can we predict the disease for a particular individual?
Yes. So that is one way, not all diseases, but if we know or if we can identify in rare diseases, as I said, the change in the genetic code. So the diseases can actually be divided into two types, majorly. One can be because there is a change in the DNA code itself. The other can be the change in the protein. Now, by sequencing the DNA, you won't get to know what changes have happened in the protein and therefore has led to the disease. But if there is a change in the DNA code, then we can actually predict. And this has been used where the infants or the babies which have still not been born have been tested for certain diseases by analyzing their genomic DNA and identifying the SNPs or the change in the DNA sequence, and can tell that what is the risk of them developing a particular disease.
Okay. Online audience, please put your questions in the chat, both in Zoom and YouTube. I don't see any other questions right now, so we can probably go to the live audience here. Yes. If there are any questions, please raise your hand and please repeat it. You're talking about RNA sequencing. So does this somehow help in the disease condition that you're talking about, like identifying different diseases? Does that help in RNA sequencing?
Okay. So okay. So the question is basically we can sequence RNA itself, and whether sequencing RNA will help in any disease condition. Yes, definitely. So if there is a change in the, as I said, it's the copying. RNA is nothing but a machinery that copies information from the DNA to protein. Now, if there is a problem in the mRNA itself or the messenger itself that the code is carrying, then you can actually identify this and also identify if the number of mRNA that are generated. So since they are shorter fragments, the amount of or the number that has been generated can also correlate to the amount of protein that is present and the function itself. So now by knowing the number of RNA that are generated or the change in the sequence of the RNA, there are some diseases which can be recognized and can be used to treat.
Okay. Question from YouTube. We just go to YouTube. We'll come back here. No worry. What is an SNP? Any comments on SNP? Is every SNP bad?
Okay. SNPs are basically single nucleotide polymorphism. SNP is single nucleotide polymorphism. To say it very simply, it's just one base difference. So for example, as I said, a DNA has a particular sequence and the main DNA has G, which needs to be present. But instead of G, it has been substituted with some other base like A or T or C. Now this change in the base, one single base, is called as single nucleotide polymorphism. So not all SNPs are bad. So as I said, the SNP, the change in the genetic code can lead to changes in the proteins itself. Now, this change in the protein can also be advantageous in certain conditions and can help us recover better. For example, a lot of allergies that are there. So in some of the cases, we can adapt ourselves by changing the genetic code and producing different kinds of proteins, or increase the efficiency of a particular protein to make them work better or function better. So not all SNPs are bad. Some of the SNPs lead to diseases, and most of the SNPs that occur in the human genome occur in the non-coding region, basically the region where the genes are not present. But there are some which do occur in the genes itself, which cause either problems or which can be beneficial or which can't do anything. They can be a neutral SNP as well.
Okay, there is another question from Joseph, which is, why do some people, in spite of having coffee, fall asleep and some don't? Is this linked to genetics or is it the coffee brand?
Okay, I don't think I'll be able to answer that completely, unfortunately. I mean, there can be some genetic basis for it, but we can't exactly. I mean, I personally do not know if there is any particular link to the brand and the kind of coffee that you are drinking. So, yeah.
Okay. Wait. There are some more, I think, serious questions.
Yeah.
What limits, Swapnil asked, what limits the length of DNA that can be sequenced using a particular sequencing method?
Yes, that's a very, very interesting question. So as I said, the read or the length of the DNA sequence that is generated in each sequencing method is different. So where does this limitation come from? For each sequencing method, it comes mostly at the detection level. So for example, when you're detecting a fluorescent molecule, and when there is a cluster of information that is forming, some of the DNA sequence can take up a fluorescent molecule faster, and in some the reaction can progress slower. So in this condition, there is a mix-up of signals that can occur, and hence we can't distinguish between one particular signal. And in this case, we terminate the sequence, or we say that we'll stop the reaction over here since we are not getting anyway the proper phase detection. So that's how, based on the detection method and based on the kind of method, there is a different length of the DNA sequence that is generated in each sequencing method.
Okay, good. Let's go back to the right audience. Any questions here? Yes, go ahead.
Yes. Okay. So the question is, how big is the data? So, yeah. Again, the data generated can also be different based on the different sequencing methods. I'll give you one example, which is Sanger sequencing and whole genomics that we did. It's around $3 billion. No, that is $3 billion. I think I am not completely sure, but it's 3 GB or no, more than 3 GB. Actually, I'm sorry, I completely forgot the amount of data, but it's a very big data, which is in gigabytes and terabytes, depending on the kind of genome that you are doing. Yeah.
Okay. Further questions?
Okay.
Okay. So let me just quickly check. There is nothing here. Anything on? Yeah, yes. You have a question. Go ahead. So I think
yes. So they do stall, they introduce more SNPs for the genetic.
So the question was the effect of non-ionizing radiation on DNA.
So these they kind of interact with the chemical base-to-base addition itself, which kind of leads to problems in binding of one nucleotide to the other, and it can also degenerate the proteins that are involved in helping this attachment to form the DNA strand. Also, it's not always that it stalls the replication completely. Sometimes it just introduces changes in the genetic code. Like instead of introducing T, it can introduce some other nucleotide, because whatever is available is just taken and attached. And this is one of the methods that is used in science to actually understand random mutations. Like they generate random mutations and see if they have any functional relevance. So,
we are mentioning about
say model organism. So what kind of benefits it will give us if we sequence different organisms?
Okay. The question is basically, we are sequencing many model organisms as well. So what is the benefit that you get? So as I said earlier, you can't do all the studies in humans, right? I mean, it's ethically also wrong, and we are humans, and we can't do all the stuff on ourselves. So what we do is we use these model organisms, which are basically having similar kind of functionality as ourselves. So by understanding how things work in a model organism, we can understand or we can correlate to the differences that are occurring in the human cell. Now, example, if we take a particular disease and treatment, we can't directly give any kind of drug to humans, right? We need some kind of initial studies to say whether the drug is actually working, whether it's harmful, or all these things. And this can't be done on humans directly. So in these cases also, we use model organisms, where first we put it on the model organism, we generate in a disease condition model organism, and give these drugs and see the effect. And thereby now we know how it works or what kind of effect it has on the organism, and thereby correlate to ourselves and utilize it for us. Yeah.
Okay. Thank you all for your questions. Will you stop sharing?
Yeah.
Okay. So thank you. Thank you for all the questions. Thank you, online audience, for your questions. Before we end the live stream, let me remind you about the session coming up. In fact, the next session, which is the first Sunday of November, 6th November at Prithvi Theatre and also online, continues on the theme of the Breakthrough Prize in Life Sciences. We will have Professor Ravi Venkatramani talk about AlphaFold, the software developments that have completely revolutionized the way we understand how proteins fold, and now can pretty much tell us the structure of every protein that we know. So for this, we will be at Prithvi Theatre 1 week from now. This is Sunday, November 6th. And remember, we're also going to play some games with amino acids in the audience. So if you're there in person, it's going to be fun. If you're watching online, well, no chai, but you can still join in for the session. With that, thank you all for coming, online audience. We can stop the live stream right now. And physical audience here, if you have a few more questions, you can please go ahead. We still have about 7 minutes, 8 minutes of time before we have to use this space for something else. So yeah, thanks again. Thank you very much, Shravani, and thanks for watching. Keep watching Chai and Why. The best ways to find out is send an email to outreach, outreach at cifr.res.in if you want to be added to our mailing list, or follow us at Chai and Why on Twitter or Facebook, and you will get the updates over there. Thank you for watching, and bye for now. That was for the online audience. You guys can still ask.